Venter is an American biotechnologist, biochemist, geneticist, and businessman. This is Dr. J. Craig Venter, who retraced much of Charles Darwin's voyage around the world. Every 200 miles of his trip, he collected and filtered seawater and found 1,800 new species and 1.2 million new genes. The purpose of this voyage was to aid in his synthetic biology project, in which he will make new synthetic bacteriophage viruses by stringing together genes in his laboratory. His new company, Synthetic Genomics, is dedicated to constructing such microbes to produce ethanol and hydrogen as alternative fuel.
Genome sequencing effort will result in key treatment for numerous diseases from diabetes to heart disease and Alzheimer's. Francis Collins and Craig Venter came together, and after almost a decade of work, Collins and Venter had essentially deciphered all the genes in the human DNA.
Craig Venter is known for leading the first draft sequence of the human genome and assembled the first team to transfect a cell with a synthetic chromosome.
Genetic engineering, the deliberate modification of the characteristics of an organism by manipulating its genetic material. Gene amplification. Gene amplification is an increase in the number of copies of a gene sequence. Cancer cells sometimes produce multiple copies of genes in response to signals from other cells or their environment. The term can also refer to polymerase chain reaction, a laboratory technique that is used by scientists to amplify the number of gene sequences in a test tube. Genetic fusion, a gene made by joining parts of two different genes.
DNA that has been formed artificially by combining constituents from different organisms.
Protoplast fusion is a type of genetic modification in plants by which two distinct species of plants are fused together to form a new hybrid plant with the characteristics of both.